Hello students, I hope you are all fine. Till now, in our all previous lectures, we have dealt with the classification system, kingdom Protista, kingdom Monera, kingdom Fungi, their general characteristic, their methods of life processes, their classification, all this we have dealt till now. But now, when we talk about the R.H. Whitaker classification, R.H. Whitaker classified so many organisms, but some cellular organism like some cellular organism like viruses, viroids, lichens, prions, they are not classified or mentioned according to the Whitaker classification. So there is also need to study this in case of classification as well, because they are commonly interacted with the different organisms like plants and animals. So if we first talk about viruses, what are they? So viruses are nothing but a small infectious non-cellular forms. They are infectious non-cellular forms. Okay, they are inert outside their specific host. They are inert, they are as it is outside their specific host, means they are host specific. Viruses are obligate parasite, they are complete parasite, they are active after infecting a host. They are inert outside, they are inactive outside, but they are active when they enter within the host. And if we talk about the structure, they are a nucleoprotein and the genetic material is infectious. They are nucleoprotein particles, means they are made up of protein and nucleic acid, but the infection material is nothing but a genetic material. As you can see, these are the diagrams where this is a diagram of tobacco-mosaic virus. This is a diagram of bacteriophage, and this is a diagram of tobacco-mosaic virus. You can see in this structure, in bacteriophage, there is a head, there is a collar, there is a sheath and tail fibers that we will discuss. But in case of TMV, that is a tobacco-mosaic virus, you can see there is a RNA nucleotides and a proteins are there. So now first we should talk about the structural component. Now in structural component, any virus is made up of three things: envelope, capsid and nucleoid. Now envelope is the outer most thin loose covering composed of proteins, lipids and carbohydrate. But these proteins are from viruses, means when virus release its infectious material within the host, the proteins are encoded by the virus genome. So we call the proteins are from viruses, but lipids and carbohydrate from the host. And this host material they use, and there is a synthesis of this envelope occurs. The small subunit known as papillomer, the small subunits are known as papillomers, and envelope is present in some of the herpes virus, HIV, vaccinia viruses, etc. And it is not present in the virus is said to be naked. So naked are those viruses in which envelope is absent. Next, inner to envelope, you will find the presence of capsid, and this capsid is a protein coat, it is a proteinaceous, and the subunits are known as capsomeres. Their number is virus specific, and this possesses an antigenic property. This has an antigenic property. So our immune response produce antibodies against this. And nucleoid, that is the virus contain either DNA or RNA. Be remember, either DNA or RNA. Here I have written or, not and. Virus contain both DNA and RNA. No virus contain both DNA and RNA. Any virus is not that which have both DNA and RNA. But in a bacteria we have studied about the mycoplasma that have both DNA and RNA. But that is a bacteria. And these are the viruses. So if we recapitulate this, we come to understand that outermost is what? Envelope. Inner to envelope, capsid. And inner to capsid, we have a genetic material. Okay. This envelope is made up of papillomers. This capsid is made up of capsomeres, and this is a nucleic acid. That nucleic acid that is a nucleotide is made up of DNA or RNA. Okay. Now, if we talk about the discovery of this viruses, name virus meaning venom or poisonous fluid that is given by Pasteur. But three scientists importantly play a role according to the NCERT, and that three scientists you should remember because they usually ask the following question from this. Now, what the scientists did? DJ Ivanovsky first came in 1892, and he recognized certain microbe as a causal organism of the mosaic disease of tobacco. What they have found? They have found that in tobacco there is a mosaic disease, colored patches are that of different color, and he recognized that for this certain microbe are responsible. And they passed these microbes from the bacterioproof filters, so they found that these are smaller than bacteria. So what they recognized? They found a microbes responsible for mosaic disease in tobacco, and these microbes are smaller than viruses. That was given by Dejevanovsky. But later on, M.W. Bezernik in 1898 showed that the extract of the infected plant of tobacco, if suppose tobacco plant is infected with these disorders, and if we remove some of the extract and cause an infection in a healthy plant, then this healthy plant will develop an infection. Means this fluid is responsible for causing the disease, that is an infectious living food, and the term for this given, that is a contagium vivum fluidum. This statement is very much important, and they directly ask this question that contagium vivum fluidum is given by. So it is given by M.W. Bezernik. And after that, W.M. Stanley came, and W.M. Stanley showed that viruses could be crystallized, and crystals consist largely of protein. Viruses could be crystallized, and crystals consist largely of proteins. That was showed by W.M. Stanley in 1935. So these three things you should remember, they are important with respect to the question. Okay. Next, when we talk about classification of viruses, Holmes in 1948 divided viruses into three groups based on their specific code. One is phytophaginae. Phytophaginae are nothing but a plant viruses. And these plant viruses generally have single-stranded RNA. They have generally plant viruses. Be remember, the genetic material of some of the viruses is fixed, either it is DNA or RNA. Phytophaginae plant viruses usually have single-stranded RNA, and the plant viruses are TMV, that is tobacco mosaic virus, potato mosaic virus, yellow vein mosaic virus, and cauliflower mosaic virus. So these are the plant viruses. Another group is zoophaginae. Zoophaginae means what? Animal viruses. And if you talk about animal viruses, they commonly have single-stranded RNA, double-stranded RNA, or double-stranded DNA. They do not have a single-stranded RNA. We remember that single-stranded RNA are not found in case of zoophaginae. And the examples are poliomyelitis virus, infectious influenza virus, smallpox virus, mumps virus, rabies virus. They are zoophaginae. Phaginae attack lower organism. In these bacteriophages are there. Bacteriophages are the bacterial viruses that usually possess double-stranded DNA, and they infect like T2, T4 lambda-phages are the bacteriophages. One term coliphase is given for the viruses which infect E. coli, and an example is coliphos FD. If cyanophase is written, means it is a phase attack blue-green algae. Blue-green algae is a cyanobacteria. Phycophages, algae, they infect algae. Mycophages for the fungi, and the zymophages for the yeast. For yeast, they have given separate term, that is a zymophages. So this is a classification of viruses. Their host, their specific genetic material, and they are a host specific, means plant viruses can infect plant only. Animal viruses will infect only. Other bacteriophages will infect bacteria only. Such that the host specificity is there. So we remember virus are host specific. That is the important point that you should remember. They are host specific. Okay. Next we talk about the another this agent like viroids. Viroids term discovered by T.O. Diener, and these are found to be a free RNA. These are found to be free RNA, means protein is absent. They are devoid of protein. They are devoid of protein coat. They are devoid of protein coat. they have RNA only, and that free RNA infectious, they are smaller than viruses, and these RNA are viroid having a low molecular weight. These viroids infect plant only, and in plant they cause some of the disease, like some 20 diseases in plant, and some of the diseases name are potato spindle virus disease, chrysanthemum plant, and citrus exocotis. You should remember this, that these are the disorders caused by the viroids. Next to viroids, prions are that. When proteins are infectious, then it is known as prions, and it is discovered by Alper et al. Now, proteinaceous infectious particles, they are prions. They cause certain disease, and they particularly cause certain disease related to the brain. They cause some of the brain disorders. So there are some disorders which are caused by them are Kuru disease, which is known as laughing death disease, bovine spongiform encephalopathy, mad cow disease, scrappy decisionship, and Creutzfeldt-Jakob disease. These are the 4 disorders which are caused by the prion. So be remember, they ask directly this question, that which of the following is the prion disorder. So protein are infectious. Next to that, there is a lichen, and lichen is a symbiotic association. But symbiotic association between what? Between algae and fungi. Now, when algae and fungi are symbiotic associated, they benefit each other, but algal component is known as phycobiont, and phycobiont are autotrophic. Yes, they can synthesize their food. But fungal component mycobiont are heterotropic. They are dependent on other. So if phycobiont is getting their food by own, but mycobiont are dependent on phycobiont, so mycobiont are getting benefit, then what mycobiont provide to the phycobiont? So phycobiont prepare a food by taking some of the elements. So algae prepare food for fungi provide shelter and absorb mineral nutrients, and water for its partner, means fungus, mycobiont provide some of the minerals and water. They are very good pollution indicator, but very good pollution indicator of what? They are SO2 pollution indicator. Now how? So we should understand some of the more things. Mycobiont is dominant partner and mostly belong to Ascomycetes. And Basidiomycetes. If fungus partner is Ascomycetes, then it is Ascolichen, and Basidiomycetes, then it's a Basidiomycetes. Example are given are there: Glaphis, Cladonia, Parmelia, Usnea, that all are Ascolichen, whereas Corolla and Cora are the Basidiomycetes. Now here the term is given as mycobiont are dominant partner. Dominant partner means in any lichen, 95% part is of fungus and 5% made by algae. Okay, so that is the thing. So because of this, mycobiont are dominant partner. Phycobiont are mostly member. Which algae are involved? So mostly chlorophyceae algae are involved, which are the Chlorella, Traboxia, and Protococcus, Parmelia, etc. Whereas it can be blue-green algae. When in spite of chlorophyceae, what other algae can be part? That is a blue-green algae, and blue-green algae are Nostochlococcus and the Cytonema. Lichen cannot tolerate air pollution. They are especially due to SO2, that is SO2 sulfur dioxide, or considered an indicator of SO2 pollution. So they are very much good SO2 pollution indicator. What SO2 do? SO2 carry out the oxidation of chlorophyll A, and due to oxidation of chlorophyll A, it convert into pheophytin. And due to oxidation of chlorophyll, what happens? This chlorophyll get reduced, and chlorophyll amount get reduced, then their photosynthesis get less. It harm ultimately algal partner, and if algal partner is lost, fungal partner is also lost. Lichens are perennial, they are slow growing. Okay, so this is with respect to the lichen. So be remember, they are SO2 pollution indicator, and this phenomena is known as pheophytization of lichen. Pheophytization of lichen means conversion of chlorophyll A into pheophytin, that you should remember. Okay, so this is all about the lichen, and that is up to this that we have discussed with respect to the biological classification, all the points, all the topics. So I hope you all are done with this. Now please student, try to subscribe this, try to subscribe channel and go with all the topic one by one. Pause somewhere where you think that you can need to study it, okay, and try to revise this again and again. Thank you.